I'd like to thank the organisers for inviting me to this conference. It's been very interesting, very diverse. Right now I'm going to go really on something very, very small, plant virus, and how you can use this particular virus for synthetic biology applications. I thought I needed a definition just to remind me what synthetic biology is all about. The key thing is, I think, is the common goal, is the design and construction of new biological functions and systems not found in nature. And my talk's going to be centered around three main aspects of this virus we're working with. It's going to talk about using virus particles, modified virus particles, for the expression of epitopes. It's going to be talking about the expression using some of this viral genome to make proteins of your desired choice. And lastly, going back to the virus again, talking about empty virus-like particles and the application they can be used in nanotechnology. So here's a brief schematic of the many roles of our virus, Cowpea Mosaic virus. As I said before, I will center about chimeras, talking about making modifications to the capsid coat protein. I will then delve into protein expression using a small component of the viral genome. And I will discuss vectors, deleted virus vectors, non-replicating vectors, making the vector system much better, development of a plasmid system. This can be used for vaccine and antibody production. I will then go back talking about virus particles, empty virus-like particles, and their role that they can play in bio-nanotechnology. So to introduce you to our virus of choice, this is a very small entity. It consists of a genome which consists of two RNA strands, single-stranded RNA strands. The first one, known as RNA-A, is approximately 6,000 bases long. The second strand, RNA-2, is 3,500 bases long. They are single-stranded RNA, not DNA. When the virus infects its natural host, Cowpea Mosaic virus, you get this lovely mosaic symptom on the leaves. If you delve in and isolate virus from those leaves, you find that the virus is distributed in three distinct groups in a centrifuge gradient. It consists of top particles, middle and bottom. The bottom two contain the viral nucleic acid. The top one happens to be empty. It's, if you think of it, it's like a byproduct of the natural viral infection. It always occurs. Key thing is you get lovely virus particles from this system, and they are very stable. And this has led to their characterization. Probably about 20 years ago, people were able to crystallize these particles and determine the atomic structure to about 3.2 angstroms resolution. And very often, we show the results of those studies as a beautiful cartoon. That cartoon consists of its coat protein, which are 60 copies of this large coat protein, sometimes shown in green and blue, or light blue, and 60 copies of the small coat protein, which occurs at the 5-fold axis of symmetry. Okay, the other essential thing about this particle, it's non-enveloped, and it's naked, if you like. That's what you see. So, as I said earlier, I'm going to talk about three aspects of how this virus has been modified over the years and its potential role in synthetic biology applications. Now, way back, I'll say way back, it's probably about 15 years ago, 20 years ago, the structure of the virus is very similar to, in the animal kingdom, foot and mouth disease virus, rhinovirus, and polio virus. So they too, like CPMV, have a 5-fold axis of symmetry, 3-fold axis of symmetry, and 2-fold axis of symmetry, and they too have a similar arrangement of coproteins. So the idea was, well, if the similarity between Cowpea Mosaic virus and these human pathogens, why not express epitopes, that's small amino acid structures that are found on the outside of poliovirus, foot and mouth disease virus, and they too may be immunogenic. So essentially, chimeras, you're making a she-goat, which is like from mythology, this is a fire-breathing creature composed of body parts of several animals. Now, a lot of work was done, but the key point to insert your favorite epitope would be on the small coat protein, where it protrudes to the outer surface. And this is known as a beta B, beta C loop that occurs in there. This is a single monomer of the coat protein, the large and the small together. And of course, there are 60 of these on a virus particle. So you would have 60 copies per virus particle of your favorite epitope. Now, a lot of work was done, and this table kind of gives an insight as to why today we are not using plant-derived vaccine for foot-and-mouth disease virus. That's just one example, because here there were 3 instances of different epitopes, 1, 2, and 3, where they were inserted into the beta B, beta C loop in a replicating virus, and unexpectedly, or it was not hoped, but the facts were that the yield was very, very small. And to overcome this, modifications were made to these epitope sequences, and amino acids were put either end, and this was a quest to get the PI of the insert down to stabilize the virus structure. This was successful to some extent, in the sense that here there were no viral symptoms on plants with the initial insert, but the addition of 2 amino acids either end, or 2 at one end, gave wild-type symptoms in the plant. The virus yield was reduced. And of course, if you want to make a vaccine to do this, you need to propagate virus, do rounds of propagation in plants over and over again, and it just wasn't practical enough. However, there is a success story actually with this approach, and if you want to vaccinate and protect mink from mink enteritis virus, it has a lovely, on the mink enteritis virus, there's a lovely 17 amino acid strand that you can actually incorporate into Cowpea Mosaic virus, and when you propagate that in plants, you will generate and then take those virus particles. You can immunize mink against the natural virus, as is shown down here. But really, there's not much cause for protecting mink unless you're a mink farmer from this virus. So this approach kind of died off, but at least it shows you can modify natural virus particles, but there are problems in the propagation and maintenance. The next idea was to use the full virus vector, and I thank Rob for his talk earlier on, because he mentioned the 2A protein from FMDV, foot-and-mouth disease virus, which is a protein that causes stuttering in translation, as he said. And some vectors were made such that the RNA2 was full-length RNA2, and at the end of this was the 2A amino acid sequence from FMDV, and then GFP. So you would then infect plants with virus from RNA1, this modified RNA2. You would hope that cleavage would occur in some instances, and you get protein production because you have the release of the protein. However, there are problems with this approach. There's a size limit of 1 kb, because principally you're relying on viral replication. For replication, you need movement in a plant. For movement in a plant, the virus is encapsulated within its capsid shell, and that moves from plant to plant through modified plasmodesmata. Plasmodesmata are modified by this movement protein gene. So if you want to put a big amount of nucleic acid in, you won't get any movement, so you won't get any production. Genetic drift and instability is a problem because you're relying on replicating virus. It's RNA, and they always make mutations, so it's not very reliable. And of course, if you're going to do experiments where you want to produce a product, you need biocontainment facilities, so you need containment glass houses. So at the end of the day, it was thought at this time in the mid-90s to delete the viral movement protein and coat proteins and see what you can get. And at the end of the day, after about 10 years' work, this led to the development of the Cowpea Mosaic virus hypertranslation expression system. So the initial experiments were to delete the movement coat protein, the large and small coat protein, and just have GFP. So essentially, we have this construct here called 1 GFP. Now, in the development of the vector system that I'm going to talk about, there were 4 key things in its development. The first development was the advent, if you like, of cloning, which of course we had DNA copies of our RNA virus, but by putting them into agrobacterium, into binary vectors, and then we're subsequently infiltrating plant leaves with the agrobacterium, this aided the infection process, and this is quite key. Another development was the introduction of an RNA suppressor protein from a related virus, tomato bushy stunt. This happens to be called P19. This would quench, if you like, host responses to viral infections and boost RNA production, exactly what a virus needs when it's replicating an RNA virus. So in this instance, you take your modified RNA2, just with your GFP expression, cloned into binary vectors with RNA1, also cloned into binary vectors, and with the RNA suppressor, you can get a lovely production of GFP protein on your leaf. It was then realized that, well, maybe you don't need RNA1 because you don't really need viral replication. Why not just see what goes on with transcription from the binary vector into the cytoplasm and then translation, see what yield you get of protein. And indeed, you don't need that. So if you leave that out, you get a result that's comparable to that. And then a follow-on from that was, well, why do we need this extended open reading frame here? So plans were afoot to remove the start codon at this position here and preserve this one here. So as you can see here, the movement protein is composed, there are two movement proteins in the Cowpea Mosaic virus. One initiates at this codon here at position 161, and then it reads through to here. But also initiation can commence at position 512. So these reading is the same reading frame. So Frank Sainsbury in our lab did some mutagenesis experiments where he eliminated the 161 and the 115. There is an initiation codon that makes a small polypeptide here, but it's not known what it does, whether you get initiation or not. And so the upstream codons were eliminated, and he tested these by infiltration of plant leaves in binary vectors, and he got this rather stunning result. So if you don't eliminate any of these upstream reading frames, you get some GFP. If you eliminate the one at 115, you get a similar amount. But if you eliminate the reading frame at 161, which is the in-frame reading frame, you get a really startling amount of GFP being made. And so why not eliminate both on the same construct? And if you run your extracts on a polyacrylamide gel, protein gel, you can see you get abundant production of a protein of the correct size. And he estimated that the levels of expression reach a gram of GFP per kilo of leaf material. If you're wondering what is going on, I'm stressed that it's translation and not the amount of RNA is present. This was a PCR experiment, quantitative PCR experiment, where the levels of the transcripts were measured in all these infections, and it's pretty constant. The graph on the left shows you the levels of GFP in these constructions, and you can see the abundant production there. So through this, RNA2 has been made hypertranslatable, and it's referred to as HT, and so the system has been known as CPMV-HT replication. So essentially, you've got 500 bases at the very 5' end of 1R of RNA2, and you've got about 180 bases at the 3' end of RNA2 that contribute to this vector. So it's an example of modifying a virus and a way for synthetic biology. So here we go. The system has a 35S promoter to make your RNA in the cell, in the nucleus, modified 5' end, 3' end, and a NOS terminator. A typical construct would look like this, between the right border and the left border, to allow you to, for the agrobacterium to transfer the plasmid into the genome, if you wish. So as I said before, hypertranslational cassette, suppressor of silencing is also there, and a selection marker if you wish to make transgenic plants, although we haven't done a lot of that kind of work. As I said before, it's agrobacterium-mediated transient expression, it's non-replicating, and these have led to the development of the peak vectors, which are small and easy to clone and versatile. So they're plasmid, easy and quick. The advantages of this system are that the very high levels of expression can be achieved within a few days. Typically we use infections for about 5-6 days. It doesn't rely on replication of an RNA virus, so we haven't got problems with size constraints or genetic drift. It's suitable for the co-expression of multiple proteins within the same cell, and I'll show you that later on. And by changing the sequences to the 5' UTR, you can modulate translation and expression. I'll also show that later on. So here's the first thing that Frank did was to use DS red, and you can see that the leaf, when you infiltrate agrobacterium, gets expressed in DS red from a peak vector. You get a lovely red glow in your leaf. If you extract that, you can see that the extract is red, and you get a large protein band on a gel. Multiple gene expression is possible. So here we have YFP and GF and CFP on separate binary vectors co-infiltrated together, but we also have them on the same binary vector, so there's one infiltration there. When you do confocal microscopy and you look down the yellow channel or the cyan channel and the merge channel, you get this lovely array of what is going on. You look at the right dilution and you can see clearly where we have YFP cells have the YFP indicator in. Where we have the CFP, you can see cells clearly infected with the CFP. So these are individual cells that either get the CFP or the YFP binary vector. But if you express both on the same vector, you get like an intermediate color here. So when you look and merge the two channels, you get like an orange color, so you get expression of both proteins. So the take-home message from that is if you want to do multiple gene expression, it's best to clone all your favorite genes on the same binary vector. So we had another student in LA, Ava Thuneman, who came in and was interested in Bluetongue disease. And a couple of years ago, this was a big problem in this country. For example, in 2008, there were no cases after an outbreak in 2007, but in France there were 28,000 cases. Now, Bluetongue virus particles consist of 4 coat proteins in varying amounts. So the viral core, which is in the middle, or on the inner layer, consists of VP3 and VP7. And then on the outside of the viral core, the outside coat proteins are put on VP5 and VP2. And of course, within a real Bluetongue virus particle, you have double-stranded RNA. Now you'll notice that the sizes of these are different, and the number of copies per genome particle also varies between them. So you need 780 copies per viral particle for VP7, but only 120 of VP3. And similarly, you need 360 copies of VP5, but only 180 of VP2. So there are 4 structural proteins equals 1,440 subunits. And it's been shown before that virus particles are efficacious and safe as a vaccine. Just to contrast the size of this particle with the one I've talked about already, Cowpea Mosaic virus, this is just a very small particle. It's only 31 nanometers in diameter. This is around 80, and there are just 60 copies of 2 coat proteins. So Ava was able to express the core-like proteins using the peak-HT system, and if you fractionate the extract from a plant down a iodixnol gradient, you can show quite clearly that you get fractions containing VP3 and VP7. If you do some transmission electron microscopy, you can see you get core-like particles. So that was very good. She then went on to co-express VP2 and 5 at the same time in the system, fractionated the extract from plants, and again you can see fractions which clearly have VP3, 2, 5 and 7. And when you look down a transmission electron microscope, you can see you get particles that look like virus-like particles. She also then played with the stoichiometry of these particles, and it turns out that you need less VP2 and you need more VP2 and VP3. So by using the HT system to express that, where in VP7 and VP5, where she didn't, you get an abundant amount of true virus-like particles. There were less core-like particles in this particular sample. Some of these particles were sent off to South Africa, and I believe that they were very good instigating and initiating an immunogenic response in sheep to Bluetongue virus. So that's an example of how you can use a small part of the viral genome to create something synthetically. There are other examples of CPMV-HT expression systems, which I haven't really talked about, though I mentioned in the abstract. And one is using antibody. You can make the light chain, heavy chain of antibodies that will come together and give you an immunological response. There's also instances of the human gastric lipase gene being expressed for colleagues across the road at the Food Institute who have a model gut system, and they're very interested in whether that can be used in their study. At the moment, they use gastric lipase that is produced from a fungal system, which is not human. Okay, the PEAK system, just to advertise a little bit here. There's been over 120 academic labs worldwide under MTA agreements who are using it. And this is all handled by PBL, and here's some of the logos of those companies that are currently using it. I don't know if they got products yet, but Megacargo, I know, are looking at human flu production. And I think they're on to stage 2 now of their efficiency testing. So I want to come back a bit more to the virus, how we can modify the virus and its potential uses. I've already talked about how genetically the outside of the particles can be modified to make for epitope presentation. But you can also chemically modify the outside, and you can also genetically and chemically modify the outside as well if you wish. So I'll give you a few examples of what's been done in the past with this. So to a chemist, they would look at the coat protein subunit and see addressable amine residues on it, and they would think, aha, I could hit that with certain chemicals. They would also look at addressable carboxylates. And some chemists over the years have performed a typical reaction where you take a dye, a Lexaflor, in this case 596, and you take some virus and you end up with coloured virus, and that is indeed what Nicole Steinmetz was able to show in the lab when she worked with us way back, and she calculated that there are up to 240 dye molecules via this amine link to CPMV. Another way is to make novel chimeras where you promote nucleation of heavy metals such as silicon oxide, iron platinum or cobalt platinum. And again, you target the B2B, B2C loop on the virus particle by insertion of an amino acid sequence that's specific, will encourage nucleation. You can perform these reactions, and over a period of time, you can then show by transmission electron microscopy, and this here is all unstained material, so the heavy atoms are incorporated into the capsid structure on the outside. So there are many other modifications you can make chemically, which I don't have time to go into. And you conclude that CPMVs can become a versatile nanoparticle. So organic, inorganic, biological molecules can be bound to it. It can be genetically and chemically manipulated. Multi-layer arrays can be structured, can be constructed, and it can serve as a template for mineralization and metallization. So it has potential roles in biomedicine as a drug delivery vehicle. However, there are problems using particles by the nature of what they are. They are viruses, they contain viral nucleic acid. And so there are several things like limits to the genetic changes you can make. They contain nucleic acid, so the regulation authorities don't really encourage it, even though it only replicates in a plant. There's no room on the inside to put anything in if you've got nucleic acid there, so they can't be loaded. So the solution is to use empty virus-like particles. Now, this is a bit of a problem because CPMV is really bomb-proof. There are some plant viruses of a similar size that you can incubate in EDTA and they will swell and the coat protein will fall off the nucleic acid and you can change the incubation conditions and the particle will reform. But you can't do that with CPMV. You can use UV to destroy the infectivity, remove it, but it doesn't remove the RNA, it's still there. And you can use high pH treatment, but you may end up with a distorted virus. So really what you would like to do is synthesize virus-free, RNA-free particles. And so what we did, we did this in insect cells, but with the development of the HT system, we just used those parts of the virus that you need to make particles. So I took the 24K protease gene from the RNA-1, which is responsible for all the cleavages in the virus, and I took the precursor to the coat protein, the large and the small, which is known as VP60, and cloned these on peak vectors. Eventually they were cloned on the same binary vector, and you infiltrate into plants and you look for virus particles. You don't get virus particles eluding from the plant on a raindrop, but this is just an image that Ava made. We took these particles up to the University of Leeds, where they do a lot of Cryo EM microscopy for us, and as you can see, their conclusions were they were 31 nanometers in diameter, both the wild-type virus and the empty particles. The empty particles are exclusively empty, as you can see in this micrograph, whereas the wild-type virus, there are some empties, namely here and here, but the rest of them have nucleic acid on the inside. When you do reconstructions, you can see that the outsides of the particle are very much identical, but when you look on the inside, the empty particles are indeed empty, whereas the wild-type viruses contain material nucleic acid. So with that in mind, why would you want to use viruses in nanotechnology? Well, they're highly regular, they are very small. This one particularly happens to be 31 nanometers. They can self-assemble, they're compatible with biological systems that you might be working with, they can be genetically and chemically manipulated, and they can be used as nanocell containers. So the advantages of making them empty are pretty evident. They're non-infectious, so no danger of spreading in the environment. They can be loaded because they haven't got anything in there, so if you can put something in, that's great. They don't have to infect, they'd have to be functional in a virological sense, so you can make radical changes. So there are new possibilities for this. And one of the avenues we're pursuing now in collaboration with others is to modify particles such that you make them magnetic and you introduce this modified particle into, say, to fight human disease against cancer. So you target your molecule to the cancer site in the body, and then you apply this small heating element to that part of the body. The particles then resonate and vibrate, and they will then kill off the cancer. That's the application here. So you need to modify the particles and load them, because you want to direct your particles to the cancer site as well, so you can maybe incorporate that. So is it possible to load virus RNA-free particles? Well, it is. This is a micrograph of unstained transmission in electron microscopy, where the particles in the left panel have been loaded with cobalt. The panel in the center here is iron oxide loaded eVLPs. And this is some verification of what was loaded. So channel 1 is just virus that has no treatment. Channel 2 with cobalt, and this reagent is specific for the detection of cobalt. Prussian blue is specific to detection of iron, of iron. So encapsulation occurs by an environmentally benign process. You can modify the external surface if you wish. So it's possible to develop virus particles or empty particles for targeted delivery. Now you may be wondering how you can load particles when I've said that they're a solid bomb-proof entity? Well, it turns out that at the 5-fold axis of symmetry, there's a tiny pore that naturally occurs in the virus. It's about 7.5 to 8 angstroms in diameter. And it is by that route, we believe, that iron or cobalt will enter the particle. However, there's a problem with this. We later then discovered that if you take fresh, empty virus-like particles and subject them to loading, they don't load. And it turns out that the small coat protein has 24 amino acids that just hang on the outside. And it was shown 15 years ago, maybe 10 years ago, that these 24 amino acids are required for suppression of RNA silencing. And over time, they fall off. So what you need to do, you need to treat your particles with chymotrypsin, and these 24 amino acids fall off, and loading can then occur. We don't know the structure of the 24 amino acids because they're structureless. So at the moment, we're working with our collaborators in Leeds to see if they can give us some data on what's going on with these ends. So I'd just like to say at the end, CPMV has been worked on for some time, probably 20 years or so, or longer. Synthetic things have been going on with the virus, and we've not really used the term synthetic biology, even though that's what we are making. The initial work with epitope presentation way back now, leading to modern things with nanotechnology and possibilities we don't know. So as I said before, we can produce novel ferrotic molecules in plants like antibodies, viral structures, nanoshells, and for potential bionanotechnology applications. I'd just like to thank everybody over the years who's been associated with George's lab, Valerie Spell and others who worked on epitopes, current group, and some who've just left working on the expression system and empty virus-like particles. And I'd like to acknowledge Dave Evans' group at the Institute who've been responsible for all the chemical modifications to particles over the years. Thank you.